Hey guys, and welcome to our Exam 2 review for lecture. So I'm going to go over the study guide, sort of like how I did in the first exam review. I'm not gonna type as much as I did last time. I'm gonna mainly point out where we can find the answers in order to answer these questions. So before we even get started, I'm gonna show you where you can find this video. So you're gonna go to modules. So everything will almost always be under modules, guys. So you're gonna be able to find it underneath the lecture one, and then our lab one that I did earlier is going to be under lab. So if you go under lecture, you'll see all the handouts that I've given out in class, as well as the blank study guide, and then the free textbook online, and then also where the video link will be whenever the recording is posted. Okay, so let's get started. So let me go ahead and pull up the study guide. So the first one is asking about what are the differences and similarities whenever you're comparing prokaryote and eukaryotic cells. So this mainly actually has to do with the handout that we kind of worked through in class, the cell activity one, where it showed a prokaryotic cell and a eukaryotic cell. So definitely be able to recognize the various um, labeling. So the labeling will be very, very similar to this. The prokaryote actually might even be the same one, just because it is a really good one. The animal cell is the one that I will probably change. I will most likely do the one that is located in the PowerPoint slides. And I don't have the PowerPoints open, so actually give me 1 second. I'm so sorry, guys. Okay, so this is chapter 4, 5, and 6. So with the prokaryotic cell, I'm almost like 99% sure I'm going to use that one for the prokaryote. And then for the plant cell and for the animal cell, I'm going to be using the following if it wants to load. So it's going to most likely look like, where are they? There it is. So it will probably be this one. So be able to label and recognize all of these components of an animal cell, as well as what are their functions. So what's the function of like the rough ER? What's the function of the smooth ER, the function of the nucleus, and so on and so forth. The only ones I am going to ask you about labeling wise will be what was on that activity sheet. So on the activity sheet, I believe it only labeled like 10 parts. So I'm only going to ask you to label the smooth ER, the nuclear envelope, the nucleus, maybe even the nucleolus, the rough ER, the ribosomes, the plasma membrane, cytoplasm, mitochondria, Golgi apparatus, lysosomes, and centrosomes. That's the only thing I'm going to ask you to label in terms of the animal cell. Whereas you see in this one, you see like the microvilli, the the peroxisomes. I'm not going to ask you to label those because that's kind of, to me, a little bit up for interpretation. So I don't really like that. But I'm definitely going to ask you the big picture organelles. So the ones that we talked about, as well as like the smoothie arm. Okay, so, and then overall, I'm going to talk about the differences real quick. So, um, and the similarities. So with the differences of a prokaryote and a eukaryotic cell are as follows. So I don't know why this is freaking out, guys. So the overall differences and similarities to a prokaryote and a eukaryotic cell is as follows. So similarities that yes, they have DNA, but what difference between them in terms of their DNA is how they're organized. So in prokaryotes, their DNA is considered circular and it's free floating. So it's not inside a nucleus, whereas in eukaryotic cells, it's a linear DNA and it is only found in the nucleus. They both have some kind of cellular membrane, so an outer um, an outer shell, if you will, that actually will separate the cell from its surrounding environment. And then also with those cellular or plasma membrane, what it does is that it acts as a selective barrier. So it acts as border patrol on whether or not something can come in and out of the cell. And then also they both have cytoplasm, which is like a jelly fluid for the cell, and then it's primarily composed of like water and some proteins. And then also they both have ribosomes, and ribosomes function to make proteins. Now, overall, their differences is mainly their how they organize their DNA, but as well as the fact of where do they store their DNA. So the biggest thing is that they do have similar organelles, but eukaryotes definitely have a lot more organelles, such as like they have like chloroplast, the Golgi body, whereas prokaryotes they're very simple where they only have mitochondria. They may have chloroplast depending on if they're photosynthetic, and then also DNA and ribosomes. That's pretty much it for prokaryotes. Whereas um, eukaryotes they have a lot more stuff going on. Okay, so that wraps up our first question. So our second question is going to be, what is the differences and similarities between an animal and a plant cell? So we've already talked about a lot of stuff about animal cells. So now let's go ahead and talk about a plant cell. So with the plant cell, I'm going to most likely have you be able to recognize this structure. So the big structure with the plant cells, I definitely want you to, of course, be able to recognize like the nucleus, the rough ER, and the smooth ER, the vacuole, Golgi apparatus, the chloroplast, and the mitochondria, as well as the cell wall and the plasmodesmatas. Those are the big picture ones. And then also I want you to know what are their functions. So why are they there? What do they serve? And within our chapter 4 PowerPoint, I definitely have a few slides that condense all of that into one. So it looks like slide 41 through 43 is pretty much where you can find all of their location as well as their function. So definitely I would look at that so you don't have to like go through all the slides because I know it could be a lot. But definitely be able to identify them, okay? So now specifically with like plant cells, since we've talked about animal cells already, plant cells themselves, the main difference between plant cell and an animal cell is the fact of a cell wall. So let me go back to that other slide to point it out. So they have a cell wall in plant cells, whereas eukaryote cells, all we have is a cellular membrane. But they do have some similarities. So the main similarities is the mitochondria, as well as some key organelles such as like the Golgi apparatus and the endoplasmic reticulum. And then also another difference is that plant cells have chloroplasts because they're photosynthetic. And animal cells, they are non-photosynthetic. So that's the big differences between them, and be able to recognize those differences if you're given a labeling portion on the test. Okay, so next what I want you to do is be able to recognize the organelles. So we've actually already dabbled on that. So number 3, we kind of already touched base on it. So it's talking about like what are the functions. Sorry guys, I should probably like scroll up for number 3. So number 3 is talking about like the functions and the organelles. So that kind of, we've already touched base on that. So let's go ahead and move on to number 4. So number 4 is also another worksheet that we did in class. So that is this worksheet. So let me make it bigger. So in this worksheet, what we did is that we talked about what was the pathway or like the steps of which like a protein was made. I'm sorry. So definitely be able to go over this and be able to put these in stepwise. Some of these you can kind of condense a little bit, which is perfectly fine, but definitely know that we start in the nucleus, and then what happens is that from the pores on the nuclear membrane, that will allow our RNA to leave the nucleus to travel to the ribosomes, and then from there, depending on which direction you're going to take, so according to our picture, it took the direction of going through the rough ER. So with that, the rough ER, it has ribosomes attached, and then from there, oh, sorry guys, what happens is that it will eventually travel to the smooth ER that doesn't have any ribosomes, and then from there it will be packaged off. And then once it's packaged off, it goes to the Golgi apparatus. So as you see right here, so like it's traveling, goes to the Golgi apparatus, and it travels in these vesicles, and then it arrives at the Golgi apparatus, and then this is where lipids are processed. And then from there, what happens is the Golgi apparatus, he will determine um, what is going to go in and what is going to go out of the cell. So depending on that, will determine if something is going to travel to the plasma membrane or if it's going to stay within the cell. So that's the general idea of how a protein travels, but I do have a whole slide about it. So let me see if I can find that slide so I can tell y'all the number. So slide 23 definitely goes over the journey of a protein a lot more condensed than our worksheet, but ideally I would like you to give a little bit more information. But this is the general picture of the journey of how a protein is made and released within or outside of a cell. Okay, so number 5, it talks about the endosymbiosis theory. So with that one, the likelihood is that what I will do, so actually let me pull up one of my other exams for y'all. So the likelihood is that I'm going to show you a question very similar to this one. So this is actually a question I give my micro kids. But I would probably give you a question of like this if it wants to work. So a question like this. So according to like the following figure, what is happening? So like which of the following statements is true? So the theory of endosymbiosis is the fact that theoretically speaking, so I'm going to go back to our figure. So theoretically, what this figure shows is that we had like pre-eukaryotic cells, so meaning that they had no mitochondria and no chloroplast. But then somewhere down the line, this pre-eukaryotic cell came along its path of either a mitochondria or a chloroplast, and then it uptook it, started utilizing it, and then it slowly diverged into the following. So definitely be able to recognize um, what is endosymbiosis and who actually came up with the theorem. So the theorem came up by Lynn. She was the one who came up with the idea that um, the mitochondria and the chloroplast were engulfed by pre-eukaryotic cells, and then they started utilizing it. So that's endosymbiosis. Okay, so the next question talks about, I believe, different tight junctions, right? Sorry, I don't know what number we're on. Are we on number 6? Okay, so it says make the table of like different cell junctions between animal and plant cells. So that mainly has to do with this table right here, if I can find it. So that refers to this table. So there are 4 different types of cellular junctions. So there's the plasmodesmata, the tight junction, the desmosomes, and the gap junctions. So with that, I would want you to be able to recognize the figure as well as their function. So that's mainly the with this table, if you can understand it and apply it, then you're good to go for number 6. Okay, so next it says name and describe the roles of the components of a fluid mosaic model. So with the fluid mosaic model, that brings us to chapter 5. So that is right here. So the fluid mosaic model, it is a phospholipid bilayer for our membrane, And then it's a mosaic model because proteins are constantly moving about within it. They're never stationary, really. And then also we have some carbohydrates located throughout the entire membrane, and then as well as cholesterol that adds the fluidity. So cholesterol, it adds the fact that it can kind of be like a constant wave like motion. So those are the main components of the fluid mosaic model. And then next question, it says explain what it means that what it means when the plasma membrane is semi-permeable. So selectively permeable, which means that it allows certain substances to cross relatively easy with little to no regulation, whereas there's other molecules that are highly regulated on whether or not it can go in and out of a cell. So that is what makes it selectively permeable, meaning that it allows only a handful amount of molecules to travel through with little to no guidance. So the type of molecules that can actively go through our plasma membrane is hydrophobic. So that means that they're nonpolar and they're small. So a lot of times, like gases can pass in and out pretty freely, such as like oxygen, nitrogen, some nitric oxide. So that can pass relatively freely. But then whenever you come to like larger molecules, they need some help, or they need higher regulation status. So that is why it's called a selectively permeable membrane. Okay, so then the next question, number 9, it says, make a table of the differences and similarities between diffusion, osmosis, and facilitated diffusion. So I don't think in the slides I created a table, but definitely, so let me go ahead and find those slides. Okay, so osmosis has to do with the diffusion of water, and it's always from a high water concentration to a low water concentration. So as you see in this picture, it shows that there's high water on this side, and then there's movement of water going this direction. Okay, so with passive transport, that includes the osmosis, the diffusion, and facilitated diffusion. All of these guys have to do with concentration gradients that are in the direction of high to low. So osmosis specifically deals with water, whereas diffusion has to do with more concentration gradient in the terms of other molecules other than water. So the movement is still from high to low, but it is pretty much any other molecule except for water. Now, facilitated diffusion is very similar to diffusion, but it needs help. So, meaning that there has to be some kind of molecule that aid in the movement where it just doesn't pass through the membrane itself. It has to go through a protein. And the example that I want you to know for facilitated diffusion is this GLUT1 transporter. So I definitely want you to know the GLUT1 transporter. That's the big one that I want you to know for facilitated diffusion. Also, with water, know also what protein it uses as well, which is aquaporins. So water can easily go in and out of the cell through osmosis, but there are times where water does need facilitated diffusion, and if it does need it, then it goes through aquaporins. Now with the GLUT1 protein transporter, I definitely want you to know the 3-step system with it. So you have glucose that will bind to the transporter, and then with that, the transporter will somehow change its shape and everything, and then what it will do is that it will close its outside port and then open into the cell, and that will release the glucose into the cell, and then with the glucose being released, the protein will then become available for binding. So definitely just know those 3 steps for GLUT1 at least. Okay, now real quick, definitely need to know the differences between hypotonic, isotonic, and hypertonic solutions. So with that, I'm going to go back 2 more slides. So hypertonic solution, this means that this cell will lose water. So water will go from inside the cell to the outside of the cell. Hypotonic solution is the exact opposite. So this is when the cell will gain water. So water will travel inside. And then isotonic is whenever there's an equal exchange of movement of water. Sometimes movement of water won't even occur because it's that perfect equilibrium where there's the same amount of water inside and outside of the cell. So with that, that brings us to this diagram. So the first part is an animal cell. So if you have too much water that goes into the cell, what can happen is that the cell can either get really, really big, but if it gets too big, it will do something that's called lysing, meaning that it burst, as you see, like it ruptured. Whereas isotonic, it'll look like a standard cell, like nothing will be out of the ordinary. Whereas if it's in a hypertonic solution, it will shrivel up and water will leave the cell. Excuse me. So that's for animal cells. But for plant cells, I definitely want you to be able to recognize that for plant cells, they normally like to be in a relatively hypotonic solution just because they like to have water constantly inside their cell because of their large central vacuole. Now, whereas if they were put into an isotonic solution, what will happen is that the plant will like start whittling and everything. It won't look as plump as it should, so your plant actually might not be as firm. So it becomes something that's called flaccid. Now, if you were to put it in a a hypertonic solution where like if you put it like in salt water when it's not supposed to be in salt water, what will happen is that the plasma membrane will actually start detaching from the cell wall and it'll undergo something called plasmolysis. And this is whenever water just rushes out of the cell and it's the plant version of shriveling. So that answers number 10 on the review. Now with number 11, it says what are the differences between active and passive transport. So the big thing is that active transport has to do with energy is necessary. So let me go ahead and pull up the next slide. So with active transport, it has to have ATP in order for it to happen. That's why it's kind of called active. Whereas passive transport, it doesn't require an energy molecule for it to occur. It will just naturally happen. And then also another difference between them is that passive transport goes from high to low, whereas active can go from low to high. So active transport is whenever you are trying to swim upstream, whereas passive transport is whenever you're just kind of going with the flow. So that will answer number 11. Now with number 12, that's our next slide. So number 12, I'm actually going to go one more down. So with number 12, it wants you to describe what is happening in the sodium potassium pump. So ideally what is happening is you are transporting out sodium molecules and then transporting in potassium molecules. So that's the big thing. And then also, like I said, ATP is required. And then also with the sodium potassium pump, I definitely want you to know the concentration of each molecule for each cell. So meaning these first two bullets right here. So these first two bullets, so the cell likes to have high salt outside. So then if it likes high salt on the outside, that means it likes high potassium on the inside. And then it's the exact opposite, the other way around. So there is low salt inside and then high potassium inside. And then there's high sodium outside, low potassium outside. So that's the idea of the sodium potassium pump. And then also know these steps. So like if I were to give you, I believe I actually gave you a picture of it on your review. Let me double check. I want to say it was a question. Or maybe I'm just losing my mind, guys. So hold on. I did. So towards the end of your exam review, I bring this up. So this is actually the sodium potassium pump in the steps minus number 6, which is where it just repeats. So definitely, like, if I were to give you this diagram, or even without the diagram, I would want you to tell me what is happening in step 1, step 2, and so on and so forth. And with that, that has to do with either this slide, which is all in one, or the 3 to follow. So either one, I definitely want you to know the steps of the sodium potassium pump, because it is pretty vital to our cell function. So with that, I think we kind of knocked out 2 birds with 1 stone with that question. So our next question was, like, define all these guys. So number 13, the big idea of why a cell has all these different components is to maintain membrane potential. Because if membrane potential is not maintained, then the cell just will most likely fall apart and then cease to exist, which we don't want that. So ideally, with this, I would want you to be able to define them. And then also, so let me pull up that slide. So with this, so this is the way that we actually maintain membrane potential. So we have a chemical and electrical forces. So the chemical force has to do with the concentration gradients, whether if it's like high or low on inside or outside of the cell, whereas membrane potential actually has to do with the voltage that is traveling across the membrane. So with the voltage, what it will do is that it will create positive and negative ions on either side of the membrane. So as you see briefly in this picture, like this side of the membrane at this point is negatively charged and this side is positively charged. And the reason why it's positively charged is because all the hydrogens are being pumped out on the outside of the cell. So that part of the cell membrane is going to be positive. So the idea is that you want to maintain that. For this scenario, you want to be able to maintain it. Sometimes you want it the other direction, but if you want to learn about that, definitely take A&P. They'll teach you all about membrane potentials. But the big picture is that you have these two working hand in hand, and then with that, they create something that's called the electrochemical gradient, and this helps drive the movement of stuff across the membrane, specifically ions itself. So that answers number 13. So I just want you to be able to apply this and then also be able to tell me the definitions of them and understand the definitions as well. Okay, so number 14, it talks about exo and endocytosis. So let's go down a few more slides. So with exocytosis, that has everything to do with that you want to move molecules outside of the cell because you want them to exit. So definitely know that with exocytosis, you have to have energy. I Oh, excuse me, guys. So you need energy in order to move any of your materials that you want outside of the cell. You have to use energy. And then normally what will happen is what will happen is that the something will travel through the Golgi apparatus, and the Golgi apparatus will package it inside the vesicle, and that vesicle will have some kind of signal telling it to go to the plasma membrane. And then once it arrives at the plasma membrane will allow it to cross so it's outside the cell. Now, whereas endocytosis, there are 3 different types that we talked about. There was phagocytosis, pinchocytosis, and cell mediated. So definitely be able to know the 3 differences of those. So phagocytosis is specifically where you are engulfing it. So think of like Pac-Man. So with phagocytosis, it's like Pac-Man going wonka 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 wonka, just eating stuff. So and a lot of times it's like food particles or it's nutrients of some sort, a lot of times with phagocytosis. Now, pintocytosis is very similar. It's a little bit on a smaller scale. And then also with the pinchocytosis, It will also suck up the liquid in the surrounding area where it makes its vesicle. And then lastly, we have the cell mediated. So this is whenever you have the cell surface receptors. This will be bound. So like right here, these are bound because they have like little purple. So I'm actually going to zoom in so you also see a little bit better. Where you have these purple components. So these receptors, the red Ys, these were bound. And the fact that they were bound, that sent a signal across the plasma membrane to be like, oh, I need to suck them in. And then he'll suck in those receptors and everything, all the ligands that he bound, and then that will travel throughout the cell. And then once that vesicle reaches its destination, then those receptors will travel back to the plasma membrane and start the process all over again. So that's number 14. So number 15 has to do what are the steps of which cell signaling occurs. So cell signaling occurs in three main stages. So we have reception, transduction, and response. So the big picture of them is at reception. This is whenever you have your ligand that binds to that transmembrane receptor protein on the cell membrane, and then from that binding will change the shape of that protein ever so slightly. And then with that, you will have transduction, which is the second part, and this is the signaling cascade of where kinase is constantly just activating proteins in a downward effect. And then with that, you will have you'll have a cellular response. So whether or not if the response is in the cytoplasm or in the nucleus will determine what happens. So if the response occurs within the nucleus, a lot of times what will happen is that will signal like protein to be made. So you'll get mRNA production, which then will go into proteins. Whereas if it occurs like within cytoplasm, then a lot of times you'll have like activation of enzymes, or the signal will go up here to be like open and close other channel ports, or even some cellular movement may occur within the cell. So depending on where the response occurs will kind of determine what will happen. So that's the big picture. Also, I definitely want you, like I said, to know this overall picture. So in this photo, it actually doesn't have one main part that I want you to know, but I'm going to draw it in real quick for y'all right now. So right here, I'm just going to color him red. So that guy right there, I'm going to change my pen color now. He's the G protein. So he's the one who does like, he will send the signal to that adenyl cyclase. So he'll send it there, and then that will signal CAMP and so on, and then protein kinase. Kind of like the question that we did today in class. So I don't remember the question actually off the top of my head. I believe it was like, what is the correct pathway of, I'll just pull up the Kahoot real quick because it is past 10 o'clock and I'm not going to remember. So let's go to Kahoot real quick and we'll just pull up that question, because I am most definitely probably going to put that on the test, guys, because it is a huge question that touches so many bases, and I definitely want y'all to know. I want you to be able to get that, because that is, if you're going to get anything from cell signaling, I want you to know that. So let's go to the Kahoot. Sorry guys, it's taking forever. Okay, so it was this one. So what was the sequence in the correct order that involves the signaling pathway using CAMP? So the correct answer is that, so before this you'll have like the ligand binding as you saw in the photo. So like you have your ligand binding, and then you have your G protein, and then your G protein goes to that other enzyme, and then cyclic AMP is activated, and then cyclic AMP activates protein kinases. So that's the whole transduction pathway. And then after protein kinases are all activated, then that is when you go into the response section. but I only wanted you to know the transduction pathway. So this is definitely a big question that will almost guarantee be on your exam. Okay, so with that, that will bring us into chapter 6 now. So that was the end of that. So chapter 6 has to do with metabolism. So with metabolism, I believe the first question was, what is the difference between catabolism and anabolism? So the main difference between them, if I can find that slide, there it is. So catabolism is cellular breakdown, meaning that you're going from big to small, whereas anabolism is the exact opposite, which is where you are going from small to big. And with catabolism, you're releasing energy. So since you're releasing energy, you're releasing heat. If you're releasing heat, it is exergonic. And then with anabolism, you require energy, so you're absorbing it, you're taking it in, so it's endergonic. So that's the big picture of catabolism and anabolism. And then also the fact that catabolism, the fact that it's exergonic, it is also considered spontaneous. And then whereas anabolism is non-spontaneous. So that's the big picture of those two reaction types. Next question, number 17, I believe. Yeah, so number 17, it asks, what is Gibbs free energy? So be able to explain to me what Gibbs free energy is. But the big thing about Gibbs is he can be defined as the usable amount of energy that is available to do work. So it's if you so think of it as like a wallet. Let's say that you want to go to the Dollar Tree and you want 3 pieces of candy, so that's $3. So that means how much work is necessary for that to happen? You need 3. So that would be our Gibbs free energy concept. That's kind of a lame example, but I think it somewhat grasps it. So it's the amount of which you can spare, essentially. And then also with that, based on certain Gibbs values, so G values, you would need to be able to determine what is considered an endogonic, exergonic, spontaneous, and non-spontaneous. So with that, I'm going to go up 2 slides, I believe. So right here. So this is pretty much all of number 17. So with exergonic reactions, they are considered spontaneous. They release energy, and then the Gibbs number, the delta G, is negative. Whereas endogonic is the exact opposite. Now, in order to calculate this, on the test I will ask you to calculate, but I will give you all the values kind of like I did here. So the equation is what is your final minus your initial, and I believe on your study guide I did put an example, so let me actually go find it so we can work it out together. Yep, right here. So right here, what I will tell you is like products and reactants, or final and initial state. So product, so if you see something called product, so actually let me make that bigger, that's really tiny. So product is your final, and then reactants is your initial. So what you would do in order to solve this, let me write it out. So remember that, ooh, that's an ugly delta. So we have delta G, oop, it is capitalized, not lowercase. So we have delta G. So we wanted our final state minus our initial state. So our final state is 750, I believe. Yes, is 750 minus our initial state, which is 1250. So then that equals a negative 500. And then kilocalories over moles. So with that, since we have a negative value, that tells us a lot. So is it endogonic or exergonic? So based on this, it is exergonic because it's negative, so exergonic. And then since it is also negative, it is catabolic. And then the fact that it is also negative, it is spontaneous. So from that, I would want you from your calculation, I would want you to be able to tell me those three components. Okay? So that answered actually number 23, 24 for y'all. So sorry guys, I think my numbers might be a little different than y'all's. I just noticed that. I'm so sorry. But either way, we're going through all the questions. Okay, so that answered. Let me zoom back out and find the question that we were actually on. So with that, yep, all Gibbs and everything. The differences between exergonic and endogonic. Okay, so now that brings us to activation energy. So what is activation energy? So activation energy has to do with, it is the minimal amount of energy that is needed to actually start a reaction. So that's activation energy. Now, with being able to define what activation energy is, you need to know the idea of enzymes. And I'm going in the wrong direction of this PowerPoint slide. Sorry, guys. What the heck? Where is it? Ha, here it is. Sorry, guys. Okay, so like with this, so activation energy has to do with how much work do I need to put in order to get my final result. So that is where a lot of times we like to use enzymes because it will minimize the amount of work that is needed to actually get to our final product that we want. So with enzymes, what you will have is actually this red line. So this is whenever you add enzymes. So the purpose of enzymes is that one, it speeds up the reaction, and then two, it also lowers activation energy. So meaning that you don't have to put as much work into it in order to still get your final product. Whereas without an enzyme, you have to put in more energy, and also it takes you a little bit longer to get to your final result that you want. So on your study guide, I believe I did put this graph on there. I definitely want you to be able to interpret it. So meaning that what is an enzyme, what's the purpose of it, and then also the fact of it doesn't alter your Gibbs free energy. So as you see right here, this kind of dark green tan looking color, it doesn't affect that. The only part that an enzyme will affect ever is the activation energy. So without an enzyme, it's higher. With an enzyme, it's lower. So be able to interpret the enzyme curve. Okay. So with that, that will bring us into actual enzymes themselves. So enzymes, theoretically speaking, they are very, very specific. They don't like to be with other things that are not them, in a way, I guess I can say. So let me pull up an image of them. So enzymes are very, very particular. They only have a certain site for a certain substrate. So a substrate is like a molecule that's floating around, and then an enzyme is a bigger protein complex. So where a substrate and an enzyme will bind is called the active site, and that active site is very, very selective. Like as you see right here, it has to be a curved side to a pointy edge. So if this was like a curve and a curve, but then the enzyme didn't match that, then no binding will occur, and then you won't get this reaction. So they're very specific. So traditionally speaking, it's kind of like a lock and key mechanism, but we now know that it's actually called an induced fit. So what actually happens, so I'm going to play a little video if it wants to work. So this is our enzyme, and then we're going to have a substrate come in, and then I'm going to explain what is happening. So you see it happened really quick. I'm going to play it one more time. So you see how this bottom part kind of comes in. So that is the induced fit. So this is whenever it kind of like clasp on to the substrate that came in. So you have your substrate, you have your active site, and then that substrate comes in, and then it becomes an induced fit. So it kind of like hugs it. So that is what is actually happening between an enzyme and a substrate. So that is how an enzyme knows that it encountered its proper substrate is because that substrate fit in perfectly and induced that induced fit. And then, like I said, the best, very simple way model of explaining it is like a lock and key mechanism. Whereas if you have, so think of your house. So if you use your home key on a door like your neighbor's house, it won't work. So your key is like the substrate, the the lock on the door is your enzyme. So if you use your home key on your neighbor's door, it won't happen. So then no induced fitting will occur. But if you use your home key on your home, then there will be an induced fit and you'll be able to actually use it. So that's kind of the very general idea of enzyme to substrate interactions. And then one last thing about enzymes is that enzymes are pretty sensitive to their environment. Their environment does affect how they work. So theoretically speaking, if you increase temperature, a lot of times that increases work, but there's only a certain threshold for them. So like if it goes past a certain temperature, an enzyme will no longer function. So some enzymes have higher optimal temperatures than others, but for the most part, if you go above 100 degrees centigrade, almost all enzymes will denature. So, meaning that they are no longer functioning. So, that's just one scenario. Another scenario is where you have pH. So, if you have like too much of an acidic environment or too much of a basic environment, the enzyme may also not be effective. So, if it's too acidic and it's a basic, it likes to be in a slightly basic environment, then it won't function. It won't do its job. Okay? So environment does affect it. So be able to tell me like two different examples and then also explain those examples. So now what if with the temperature one at least, what if we went colder? So it has the same idea. The only difference is that the enzyme may not um, quote unquote denature. What may happen is that it'll become somewhat dormant. It may denature depending on how cold it is, but for the most part, it'll just become inactive. And then sometimes you can heat it back up and put it back in its optimal temperature, and it'll still function. It may not function to 100%. It may only do like 85% um, productivity, but it can still function. But definitely know the concept of high temperature, low temperature, and then pH values on how does that affect enzyme productivity or work. Okay, so let's see where that brings us. Um, enzyme activators and inhibitors. Okay, so one last thing on enzymes, and then I'm going to go over the labeling, and I believe at that point that will conclude our review. So with enzyme activators, it is they are non-proteins and they bind to the enzyme or they bind to the substrate. So with that, what will happen is that a lot of times um, with them binding to the enzyme or the substrate, it allows the enzyme to know that it needs to do work. So let me just show you. So what I mean is that if you don't have, some enzymes need helpers, so these helpers are called coenzymes. Actually, I'm going to change that color because that's an ugly color. Let's go with pink because pink is pretty. Well, pretty-ish. So what will happen is that so you see right here, um, this molecule doesn't look like it fits the active site. So the fact that it doesn't fit the active site properly, no binding can occur. And then if the enzyme and the substrate don't bind together, then there will be no reaction. So in that case, it needs an activator, meaning that it needs a helper. So this coenzyme will come in and bind to part of the enzyme, and then with that, the actual substrate can come in and fit very, very nicely, and then induced fit will happen, and then the reaction will occur. So with enzyme activators or coenzymes, what will happen is that it gives a signal to the enzyme that, hey, you need to bind a substrate so you can do the reaction. Whereas an inhibitor is the exact opposite. So an inhibitor, what it does is that it will inhibit or block the action of enzymes, meaning that it doesn't want the enzyme to do work. And there's two different ways. So the first way is called competitive, and then the second way is non-competitive. So competitive is where something will actually bind to the active site, blocking the substrate. So that is competitive, where they're actively competing for that active site where the substrate normally likes to bind. Or there's non-competitive, which is where a inhibitor molecule will bind to a different region on the enzyme. But once that non-competitor inhibitor binds, what it will do is that it will actually change the conformation or change the shape of the active site. And with the active site now different, that original substrate can no longer bind because it's not the proper shape. It's like trying to put a round peg in a square hole. It just won't happen. So that's the idea with enzymes. It has to be like circle to circle, square to square. It can't be circle to square. Okay? So that's the big picture with enzymes. And like I said, I think with that, it brings us to our labeling. So with our labeling, I'm going to actually pull up, yep. So like I said, with the prokaryote, I'm going to pretty much show you the prokaryote. Don't worry, guys. On the test, it will be in color. I promise. And then also, I will definitely have the little label lines a lot more defined. And with that, they're going to be maybe, oops, something similar to this. Okay, so they're going to be something very similar to this. So like I said, the prokaryote, this is probably what you're going to see on the prokaryote side. And I'm going to fix that. Now with the animal one, I'm going to show you the one that was in chapter 4, and then I'm going to only have you label the big guys. So like the mitochondria, the centrosomes, the smooth ER, the rough ER, Golgi apparatus, cellular membrane, the nucleus and the nucleolus, and then the nuclear membrane or envelope. And then also with the plant cell. So I kind of already highlighted that. So definitely like the Golgi apparatus, the nucleus, the rough ER, the smooth ER, the vacuole, mitochondria, cellular membrane, plasmodesmatas, cell wall, and then chloroplast. And with all those, definitely know the function. Okay, guys? So, like I said, I believe that concludes our review session. Let me double check. Oh, one more. Membranes. Good thing I double checked, huh, guys? Okay, so with plasma membranes, it won't be, I won't do shapes like this, but I kind of just wanted to show you. So these are the big ones that I will test for. So definitely know the components of it. On the exam, it will be in color, like I said. And then what will happen is that I'll probably have lines with letters and be like, what is letter A? And so on and so forth. Or what's number 2? I don't know. I don't know if I'll be in a number or lettering mood. But be able to recognize these components. So definitely pause the video, look at this, see what's going on, okay, guys? So that wraps up our review session. So I really hope this helps you guys. If you have any questions, definitely don't hesitate to ask me. Um, Email me, ask me in person, set up a meeting, whatever it may be. Also, go to the tutoring lab that is right below our classroom. They are extremely helpful. Maybe they can even give you some study tips and so on. But if there's nothing else, guys, I will see you in class and happy studying.